Hello friends, welcome to Sam Biology class. Today we shall discuss about the structure of bacterial promoters. Before that, I request all of my friends, please subscribe my YouTube channel and watch and like my videos and also share them with your friends. A promoter is a DNA sequence to which RNA polymerase binds to initiate the transcription of a gene. In transcription, out of two DNA strands, only one is used as a template on which RNA is synthesized. This strand is called template strand or non-coding strand or the antisense strand. The other strand is non-template strand or the coding strand or the sense strand. Both the strands run in opposite direction. The template strand runs in 3' to 5' direction, whereas the non-template strand runs in 5' to 3' direction. In this figure, +1 is the transcription start site from which transcription starts and proceeds in 5' to 3' direction. It is the nucleotide pair that corresponds to the first or 5' nucleotide of the RNA transcript. The nucleotide sequences, those are located to the left of the transcription start site, are called upstream sequences, and those located to the right are downstream sequences. Nucleotides or nucleotide pairs located within the upstream sequences are given minus prefixes, and those within the downstream are given plus. Promoters are usually located near the transcription start site, typically upstream of the non-template strand. They can be 100 to 1000 base pairs long. Hundreds of E. coli promoters have been sequenced and are known to have surprisingly little in common. This is a bacterial promoter to which RNA polymerase binds to initiate the transcription of a gene. Bacterial promoters contain two conserved sequences. One is the -10 sequence and the other is the -35 sequence. -10 sequence is also called Pripnobox or Pripnoscalar box. It is the bacterial homologue of the Tata box. The midpoint of -10 sequence occur at about 10 nucleotide pair, and that of -35 sequence occur at about 35 nucleotide pair before the transcription start site. Although these sequences vary slightly from gene to gene, some nucleotides are highly conserved. The nucleotide sequences of these two conserved genetic elements are known as the consensus sequence. The -10 consensus sequence in the non-template strand is TATA-AT, and that of -35 sequence is TTGA-CA. The sigma factor initially recognizes and binds to the -35 sequence. Hence, it is also called the recognition sequence. The AT-rich -10 sequence facilitates the localized unwinding of DNA around the transcription start site that result in the formation of transcription bubble. The transcription bubble is a molecular structure in which the two DNA strands are separated. It enables the template strand to base pair with the incoming ribonucleotides. The distance between -10 and -35 sequence in E. coli is highly conserved. It is never less than 15 or more than 20 nucleotide pairs long. In addition, the first or the 5' base of the RNA transcript in E. coli is usually a purine. In addition to -10 and -35 sequence, an additional DA element that binds RNA polymerase is found in some strong promoters, for example those directing the expression of ribosomal RNA genes. This additional sequence is called up element, which is located upstream of the -35 sequence. The up element increases the RNA polymerase binding by providing additional specific interactions between the enzyme and the DNA. Another class of sigma 70 promoters do not have the -35 sequence. In such cases, the standard -10 region is extended at its upstream end. This additional sequence is called extended -10, which provide additional contact between the enzyme and the DNA, thereby compensating for the absence of the -35 sequence. So friends, this is all about bacterial promoters. For more updates, subscribe my YouTube channel, Sam Biology Class.